Assalamu alaikum. I am Dr. Tijani Ahmad, not Hassan. Hassan, he is not here because he is on duty outside of the country. I will present BBT instead of Hassan, inshallah. I am from DAFSA, Biosecurity Affairs Division, pathologist expert in Al-Zafra region. Meanwhile, the presentation is be seen. I will give a brief about our reference laboratory, our collaborating center, WUHA Collaborating Center, who did this success. It is an international collaborating center designated by WUHA or Animal Health Organization in May 2022. As a reference or collaborating center for camel diseases. It's hosted by DAFSA in Abu Dhabi. The center is mandated to do various activities, including diagnostic of camel diseases, and as well as another animals producing food animals as well, and also participate in zoonosis risk assessment as part of One Health approach. In addition, the center gives technical support and consultation to WUHA and its countries. This is our outline or summary of presentation about WUHA and overview of the collaboration. Selected previous literature review about the condition of UCD or unknown camel disease, UCD field visit, and collection of EpiData, sample collection processing, investigation strategy, major laboratory outcomes, capacity building at AHI or at Ethiopia laboratories, limitation and way forward. As I said, WUHA Collaborating Center is one of the expertise center for camel diseases designated by World Organization for Animal Health in May 2022. The scope and activity of the center to cover diagnostics of camel diseases and conducting epidemiological survey, pathogen characterization using latest technologies. Also participate in capacity building of animal health and laboratories in the regions and elsewhere, contributing in global One Health approach. And our laboratory also has many designations in as National Reference Laboratory for CCHF and MERS-CoV, and National AMR Consortium members, as well WUHA Coronavirus Network member as reference. WUHA Collaborating Center falls under Biosecurity Affairs Division, which consists of 5 sections, regional laboratories, biobank section, biosecurity, and a risk assessment section, reference laboratory section, and technical support. Under reference laboratory sections is WUHA Collaborating Center for Camel Diseases, located in Abu Dhabi or Al-Wasba Veterinary Laboratories. Our laboratory, highly equipped with latest diagnostic technologies, enable us for diagnosis of unique problems in Ethiopia, and also here we participated in several diagnostic issues in the national levels. Due to our highly equipment with latest generation equipment like sequencer 1, 2, and 3 generation, or Sanger sequencer machines equipped with bioinformatic programs. Overview of collaborations. In 2024, DAFSA received official contact from Ethiopia Agriculture Authority about possibility of participate or support on investigation of unknown camel disease. What is unknown camel disease? It is a fatal disease affect camel in the Horn of Africa for long times. But the actual cause of the disease is not identified, and this issue is very serious and cause severe economical loss in the area. Upon that contact or official letter, there is several meetings were held in cooperation with FAO and WUHA members and Ethiopian Agriculture Authority, and discuss the issue and recommend further analysis of or investigation of the problem. ADAFSA nominated an expert teams from different specialty headed by Dr. Muhammad Al-Breki, reference laboratory section head and member of Prof. Abdul Malik, virologist, Dr. Hassan Zakaria, molecular biologist, Dr. Tijani, pathologist, and Dr. Noura, pathologist. Before our traveling, we're reviewing the problem to see what is the researches conducted regarding the problems. We found several researches carried out, but none of them indicate any pathogens. Most studies incriminate viruses as the cause of the mortalities, but no virus were identified. These all researches conducted, but no virus was identified or any other pathogen except BBR detected as in serology as antibodies and antigen, but not isolated. Joint field visit, after we went to Ethiopia, we met a team from Ethiopia, from Ministry of Agriculture, and some colleagues from regional veterinary laboratory at the field of or the affected area, Yabello area. We conducted a short visit for 3 days. During the visit, we met the local farmers to take a comprehensive case history of the disease, to know the behavior of the disease, to collect sample from clinical sick, from recovered animal, and also we did a postmortem to collect tissue from sick and dead animals. Several samples were collected, about 117 samples, swab, blood, tissue, fecal, tick, and blood smears were prepared at the field, and samples were prepared for shipping. At AHI, samples were allocated into 2 batches. One keep there as archived or backup samples, and the other batch send it to UAE for analysis. ADAFSA investigation, upon receiving the samples were distributed to different laboratory units for analysis. This is the major findings at postmortem. When we did a postmortem of sick camels, we found the affected organ, heart and intestine, lymph node, and kidneys. This is the main affected organs, and we collect panel of sample from all affected tissue in as fresh sample and formalin fixed sample for histopathological analysis. Samples were also allocated based on plan of diagnosis. Sample for molecular, sample for virology, isolation, sample for histopathology, parasitology, and all discipline of diagnostic. Histopathological analysis revealed some parasitic infestation at the intestine. We see in the first slide, we see this red color is stage of coccidium. It is an opportunistic pathogen, need a trigger causes, just like virus or any stress factor. This can initiate the infection by coccidiosis. Another slide shows verminous enteritis. This larvae of a worm, internal parasite, larval stage of strongyloids, also concurrently emerged with other diseases and it also causes immunosuppression. This intestinal section, we see black dots or blue dots indicate for cellular infiltration or cellular immune response. In viral infections, the mainly cells infiltrated is lymphocytes. We can differentiate type of inflammatory cells into lymphocyte, neutrophils, and accordingly we can suspect the etiology of this immune response. The heart also show necrotic changes with infiltration of lymphocytes, and other section revealed evidence of sarcocyst, one of zoonotic diseases, but it can found not the cause of the disease, but can co-infection. Parasitological analysis of the fecal samples revealed mixed infection of internal parasite, Emeria, Trichostrongylus, Trichourus X. 9 samples of ticks classified into Hyaloma, Rhizocephalus, and Bilioma species. Acid-fast staining smear from fecal sample revealed negative for Cryptosporidium oocyst. Gene sustained of blood parasites, 17 samples, also no blood parasite was detected in the smear. Bacteriological analysis revealed isolation of various species of bacteria, most of them classified as environmental because we did postmortem in the field. And this lead to high contamination of the sample. So the isolated bacteria classified mostly as environmental contaminants. Virology, no virus was isolated because for long period of preservation and shipping. This may affect the viability of the virus, so we cannot isolate the virus from any of the samples. Molecular analysis. In our molecular approach, we did pan analysis or PCR analysis targeting the genus level and family or subfamily level, and then we did a sequencing. If we find any positive result, we did a sequence to identify the possible etiology. These are bacteriological PCR analysis. All samples tested against Pasteurella, Mannheimia, and those bacteria and gave negative result. All samples tested for pan-trips blood sample and some tissue sample also negative for Trypanosoma evansii. Only one sample reacted positive for Salaria species, and one sample reacted positive for Anaplasma species. All tick screened for CCHF, Q fever, and Borrelia species and reacted negative. Another approach was followed was pan real-time PCR assays targeting family of the major viruses in the samples. We did pan blue tongue virus, pan bovine viral diarrhea virus, pan corona, and pan paromyxo, Rift Valley, foot enzootic, Raymond, camel box. Only pan flavivirus was found positive, gave positive reaction in 18 sample of tissue sample out of 35, and 3 swab sample out of 32 samples were positive, and no blood sample was negative. All blood sample were negative. The positive sample further identified using sequencing and bioinformatic tools. And the detected virus was identified as Wesselsbron virus. Based on our findings, we activate a direct real-time PCR test and retest all our sample again using our new activated test. And based on this, 25 instead of 18, before 18 positive, now tissue sample 25 positive. Blood sample, before no sample was positive, now three sample were positive. Swab sample remained the same as pan flavivirus real-time PCR result, not affected. Means the sensitivity of the test is increased. Our sequence, partial sequence, deposited in the gene bank as the first sequence isolated or identified in camels. No any data regarding Wesselsbron virus or sequence of Wesselsbron virus from camel were found or deposited in the gene bank before. This is the first time identified this virus in camels and the first deposition of the sequence in the gene bank. The virus was further characterized using phylogenetic analysis to know which clade is our virus. The virus has two clades, clade 1 and clade 2. Our virus ranked under clade 1, which is known as more pathogenic clade than clade 2. And in the lower part of the section, we saw the red color indicate the number of tissue reacted positive for the virus, and this somewhat indicate the tropism of the virus to a special organ or system of the animal, and this we can correlate it with the clinical samples of the sick animal in the field. This helps us to select sample from the field instead of necropsy if we find clinical affected animals. Our results were finalized as article and published in a highly impact journal, Emerging Infectious Disease, Molecular Detection of Wesselsbron Virus in Dromedary Camels. This background about the virus, Wesselsbron virus is a mosquito-borne virus, belong to Flaviviridae. The virus was first isolated in South Africa in the district Wesselsbron districts in 1955. Later, the virus isolated from febrile human and mosquito, also in South Africa, and this proved it is zoonotic potential. Later, the virus reported in most African countries. The virus is known to infect mainly sheep and cause abortion and teratogenic effects of the fetuses. Also report in equine cause nervous signs. The virus cause nervous signs in equine and report to infect human as zoonotic potential. It is importance as public health risk. The result of this investigation opens the door for further research into Wesselsbron virus research in camels, while the role of other pathogens cannot be ignored, because this very short period of survey, and we just targeted a small geographical area. We need to expand the survey in other regions to know the distribution. Also, the isolation and experimental infection is necessary to prove its role in pathogenicity in camel. The virus has a public health importance, and this is the necessary point. The data are shared in various conferences in Ethiopia and in national levels. Capacity building also carried out while participating in Ethiopia by activation the same test in Ethiopia laboratory, a retested of archived sample, and hopefully we find 8 samples from archived sample were positive, and this support our finding also because the sample collected from different area and different season. Lesson learned, major limitation, logistic, like postmortem of animal expense we have to pay if we want to do postmortem, sampling, you have to collect panel of samples. Also, shipping, we have to plan for shipping in previous multidisciplinary sample and analysis also for different sample from different animal, from human to study the zoonotic potential, the effect of the disease for other animals. This conclusion. Our study provides in evidence of Wesselsbron in various tissues and swabs, detection of virus in the necropsy tissue, support the infection and distribution. Clade 1 virus is the most recognized as pathogenic. Partial sequence derived from camels and expanded the known of geographical range. Further research, including whole genome sequence, pathogenicity, and experimental infection is needed. Thank you.
Thank you so much, Dr. El-Tijani, for highlighting the latest evidence and molecular technology to address the virus in camels in Ethiopia. We have room for one question, if there is one.
Yeah. Thank you very much for the phenomenal work that you've done. I just may have missed the species of mosquito that is actually carrying the virus. Is it known which species of mosquito is transmitting it? Is Aedes aegypti or?
Aedes species, I don't know exactly, but Aedes species.
Aedes species, okay, thank you. Thank you very much.
Thank you so much again, Dr.
The fact that we actually missed to collect the mosquito. And this is what we are showing here in this comprehensive plan. The comprehensive plan, one of the targets as well we need to collect is the mosquito, to make sure which mosquito or which subspecies is involved in this.
Thank you so much, Dr. El-Tijani, again.